Hi, everyone. I want to thank you all for joining us today for our second webinar for our patient involvement group. So today's webinar is called Emerging Therapeutics, and we're going to be talking about some of the milestones that have come up in the history of cancer and in the development of therapeutics, as well as where we currently stand today and kind of what we're looking for in the future as we move forward with new therapeutic interventions. Next slide. So for those of you who were not able to join us for the first webinar, I'm just going to really briefly introduce myself. So I won't read my whole bio here. You're welcome to read that on your own time. But basically, I'm currently doing my PhD, and I'm working in immunotherapies for breast cancer. And I also am a 7-year survivor of triple-negative breast cancer. So I'm fortunate to have a very unique perspective as a researcher and patient in the same field. And I'm really happy to be able to work with you all and talk to you and hopefully give you a bit more perspective about the way that we look at cancer from a researcher's perspective as well. Next slide. So I chose to start off with this slide before we delve into therapeutics because I think that as we come into the era of social media and the era of fake news, it is very easy for real science to get lost in the media and just kind of get lost in the muddle of things. And science communication is more important now than ever. So unfortunately, scientists historically have not been great at really presenting our science to anyone other than scientists. But there is a really big push right now for scientists to start to work with more advocacy groups and to work on lay terminology for the work they're doing so that they can actually explain to people who do not work directly in their field what they're doing and why it's important. And I work with the Canadian Cancer Society a lot. I head up their research information outreach team here in Hamilton. And our sole purpose of that group is really research dissemination to the general public. And I can't stress enough how important it is because everything that we do in the lab ultimately means nothing if we can't tell people about it and educate people about it so that they feel comfortable and feel that they're aware of the progress that's being made with everything that we're doing and everything that they're donating their money and their time toward. Next slide. So starting all the way back with the very first cancer treatments. So cancer really has been discovered so long ago, it's kind of crazy to think about, but it really came about all the way back to the development of the scientific method by Galileo and Newton. And we had the first autopsies even as early as 1628 by a scientist clinician named Harvey. And when we talk about the first therapies for cancer or the first treatments for cancer, what we're really talking about is kind of these raw surgical procedures that were being done. And the famous Scottish surgeon John Hunter, back in the 1700s, is the first who suggested that some cancers could actually be cured by surgical intervention. And he really described how the surgeon should decide whether or not to operate based on whether or not the tumor had invaded nearby tissue or not. And this is the first time that we start to see this delineation where people are looking at whether or not cancer has metastasized and correlating that to whether or not they're going to do well with surgical intervention. Next slide.
So the birth of scientific oncology really came when we had the use of the first microscope. So this was the first time we were able to actually look at tissues more closely and see differentiation between cancer cells and normal cells. And this really paved the way for modern day pathology and how we began to classify and look at different cancer types. And, you know, now we really have that we can actually have a diagnosis instead of just having these lumps that were considered to be all kinds of various things from different doctors. I think, you know, you could have one lump discernibly said to be one thing by one doctor and something different from somebody else. So this is the first time where we started to see more precise diagnoses being made for cancer patients. Next slide. So like I said, surgical progression was really primitive in the early days. So, you know, it progressed much better for patients once we had that discovery of anesthesia. Of course, now patients could endure procedures a lot better. And then we saw the era of the radical mastectomy. So William Halsted is, you know, the famed breast surgeon who developed the idea that instead of just removing cancerous tissue from a breast, we really should be trying to remove the margins, which is a concept we still use to this day, but he took it to the extreme where he was not only removing a patient's breast, but he was also removing, their pectoral muscle and their clavicle and all of their lymph nodes, and really disfiguring these patients. So even if they managed to overcome the disease, most of them were left with pretty severe complications from the surgery itself. And really, the radical mastectomy stayed in place for almost 100 years. We really saw this as the century of the surgeon. Next slide. So in the late 1800s, we have Marie and Pierre Curie discovered the radioactive elements of radium and polonium. And this was a massive discovery. So now we start to see basically in the invention of radiation that we can now use not only radioactive isotopes, but also, this concept of just radiation in general being able to treat cancer. And of course, this paved the way for modern day radiation therapy and radiotherapy. But unfortunately, at the time, what they didn't realize is that radiation can also cause cancer, of course. So we had people that were handling these things in their bare hands and being exposed to large doses of radiation and radioactive substances. And, of course, Marie Curie ended up passing away actually from cancer that she contracted from handling all of this radioactivity. Next slide. So the early 1900s, it seems crazy. It's been more than 100 years since the immune system was really thought of as playing a role in cancer. And we are only now really starting to see people investigating this claim much further and just trying to harness that immune system. So it was Paul Ehrlich that originally proposed that the immune system suppresses tumor formations, that it plays a role in actually evicting these cancer cells from your body. And this became known as the immune surveillance hypothesis. And, this is something that we still discuss in research to this day. It's still very relevant to this day, and we'll see that as we get a bit further through the slide deck here. Next slide. So this is a very famous picture. So in World War I, of course, nitrogen mustard gases were weaponized and used in huge amounts. And it was really doctors started seeing these soldiers coming back from war that had these really depleted bone marrows, and they couldn't figure out what was going on with these patients. And it, of course, was because they had been exposed to these large doses of nitrogen mustard gases. And so finally, in the mid-1900s, after a lot of research and a couple people really investigating what was happening at the cellular level from these gases, nitrogen mustard was actually approved finally for the treatment of cancer, of course, in much lower doses than what had been used for the weaponized version. Next slide. So in 1953, we had the first complete cure. So Roy Hertz and Min-Chu Lee achieved this cure in a solid tumor. So they gave this patient methotrexate. And what was interesting about this case was that this was actually the first instance when they decided after the disappearance of the tumor that they were going to actually keep treating the patient with methotrexate, because this was proposed by Dr. Lee that perhaps there were cancerous cells left in the body, that if we kept treating the patient even after the symptoms and the appearance of the tumor was gone, that perhaps we could prevent relapse. And at the time, that was a really crazy theory to be postulating, and people thought that he was crazy. He actually pretty much lost his job and his career for treating this patient so extensively with methotrexate. But of course, in the end, he saved that patient's life and really paved the way for how we think about treatments nowadays, where we really do treat patients long after the disease is gone, at least visually. Next slide. So combination chemotherapy came on pretty fast and pretty heavy. So it became pretty rapidly apparent to clinicians that a lot of people are resistant to chemotherapy. And only when we give chemo in combination do we see much better rates of survival, and we see much better efficacy from the drugs. And this is largely due to the fact that chemotherapy comes in a wide array of different types of drugs. So if a patient is resistant to one type, it's highly likely that they will benefit from a different type. And when we saw combination chemotherapy first coming into the clinic, the thought process was usually that the higher the dose, the better. So they really didn't think about the side effects to the patient and the fact that, ablating these patients with severely high doses of these combination chemo regimens actually predisposed them to get other types of cancer later in life. Next slide. So breast cancer is one of the subtypes of cancer where we've really seen the most clinical progress from research. So, we had the approval of tamoxifen in the 1970s. That was really the first anti-estrogen drug that was developed for the treatment of breast cancer. It was originally discovered as a birth control treatment. It ended up not working for birth control. So this drug actually kind of sat in the world of medicine for many years and thought to be a useless drug and no real use for it before someone finally discovered that it could be used for the treatment of estrogen-positive breast cancers. Excuse me. Then we had the HER2 oncogene identified. Oncogenes are actually a pretty relatively new discovery. It was only really the early 1980s when we started to think about these cancer oncogenes, how their overexpression can affect the growth of different cancers. 1985, about 100 years after we saw the radical mastectomy coming down from William Halsted, we finally had clinical data supporting that women with early stage breast cancer could be treated with a lumpectomy followed by radiation, and they had really similar rates of overall survival than they would have had if they had these more radical invasive mastectomy procedures. Late 1990s, again, we have another drug that's approved for the estrogen receptor positive advanced breast cancer, and this is when we really started to get into the time of various hormonal therapies coming down the pipeline. Next slide. So, of course, most of you, I'm sure, or all of you know of the HPV vaccine. So there was theories for a long time about various types of viruses being able to cause and induce cancer, but HPV is really the first one that we saw where we had been able to discover the virus and develop a vaccine that was actually going to be preventative in nature. And that really revolutionized our current thinking of what cancer vaccines could look like in the future. And unfortunately, when this came out, I think a lot of people in the media portrayed it as we're going to have a universal cancer vaccine, which really could not be further from the truth. But I think it's great to know that for certain types of cancer, it really is possible to really harness the exact origin of that cancer, or something that really puts people at a much higher predisposition for getting it, and we really can have these highly efficacious preventative vaccines. Next slide. So taking a different switch here, what goes into determining your treatment? So of course, there are so many different things that an oncologist has to think about when a patient presents with cancer. As you all know, there's so many different types of cancer and so many subtypes within each of those types that each patient presents with a really unique individual case. Even two patients that have the same type of breast cancer are going to have different treatments based on the type of cancer, the stage of the cancer, their personal preferences. One patient who's 20 may opt for a treatment that could perhaps preserve her fertility versus another patient who's 50 who doesn't care whether she becomes infertile or not. So all of these different things, your age and your genetic factors, these all come into play when an oncologist is trying to go into determining your treatment regimen. Next slide. And what are the different treatment plans? So we have your first line therapy. These are, of course, the most common and standard treatments that are given for any type of cancer. They vary, of course, between the types of cancer. We can have neoadjuvant therapy, so it's given prior to other therapies. So this is a good example of this, is breast patients nowadays are often given chemotherapy prior to surgery in order to shrink that tumor before it's removed, and also to really gauge whether or not that patient responds to the chemotherapy anyhow, as there's really no way to know that after you've already removed the primary lesion. Alternatively, we have adjuvant therapy. So this would be the instance of chemo being given after surgery. So this is really given to destroy any cancer cells that are left behind and reduce your risk of recurrence, or that it will reoccur, sorry. Next slide. Your treatment goals can be different, and of course they're different based on the individual situation. So again, we can have prevention. We can talk about something like the HPV vaccine. We can have a cure, so we're looking to really cure this patient. We think they have a high likelihood of progressing tumor-free and actually getting rid of all of those cells in their body. Just to control the tumor, sometimes, we really know that it's not a curative disease, but we really can have a really good control of that tumor and give that patient good extended quality of life. And then, of course, there are palliative cases when the treatment goal really will be palliative care to increase the quality of life, relieve pain, and just really ease the symptoms that that patient is feeling. Next slide. So the types of treatment can be local, systemic, or targeted. So we can either give it directly to the primary tumor itself, we can deliver it through the bloodstream so it travels through the entire body, or we can use drugs that we can deliver systemically, but they go directly to the tumor. So those are targeted to certain molecules or proteins that will be highly overexpressed in the tumor. So it's like we inject this little lock into the body and it goes and travels through the bloodstream until it finds the proper key. Next slide. So what treatments are available nowadays? So coming out of, kind of the raw and horrific ages of cancer exploration and discovery, where are we at now? Next slide. So of course, surgery. Surgery is and always has been the #1 intervention for cancer patients. So we can use it to prevent cancer, diagnose and stage cancer, remove cancer, relieve symptoms, lower the chance of recurrence, repair damaged tissue, or just to support other treatments and bodily functions. It's often used as part of a treatment plan in conjunction with other therapies such as chemo or radiation. The con is that, of course, some tumors are not operable, depending on where they are and how sensitive the surrounding tissue may be or how widely they may have spread. And some tumors present with really difficult tumor margins. So even if we remove most of the mass of the tumor, it's really difficult to excise all of the cancerous cells. And that's why we typically use it in conjunction with other therapies as well. Next slide. So more recently, we've seen the emergence of robot-assisted surgery. So this allows doctors to perform really complex surgeries with much more precision, flexibility, and control that we possibly would have seen with conventional techniques. It's usually associated with minimally invasive surgery because usually this can be done through small arthroscopic incisions, and we don't need to have huge open incisions for the patient. But I will say robot-assisted surgeries have really come of their age for certain conditions. So especially, gynecologic and cardiac procedures have really started utilizing this in high amounts, and it's been really supported by the FDA and by various organizations to do so. But it also is very expensive, and it's actually not currently approved for cancer surgery because there are concerns about the outcomes. So there's a lot of research going into specifically using this type of surgery for cancer right now. But like I said, it's kind of a controversial area. And, we're not talking about just stitching somebody up. We're talking about a much more complex disease, a much more complex operation, where I think it's a bit controversial as to whether or not you need that eye right in there to get the proper margins anyhow. Next slide. So radiation therapy, just as kind of a generic slide here for it, radiation works by destroying the cancer cells and damaging a cancer cell's DNA so that it can stop dividing and growing. So it can either shrink or completely destroy a tumor, and it is most effective on rapidly dividing cells. So it will be most effective on cancer cells. But with that being said, it is not specific, and it will cause damage to some normal cells as well. Next slide. So there are many different types of radiation therapy nowadays, and really keeping in mind that con from the last slide, that radiation therapy is not specific and it can cause irreversible damage to healthy tissue as well. All of these different types of radiation therapy have now come into play to really try to get it more focused so that we're really decreasing the amount of damage done to healthy tissue. So we have intensity modulated radiotherapy. This is, again, really focusing on delivering precise radiation to the tumor to minimize the dose to surrounding normal tissue. Image-guided radiation therapy, I think, is really cool. It uses imaging at the same time that the radiation is being delivered so that we can improve the precision and accuracy. But it's actually usually used to treat areas of the body that move. So, for instance, the lungs. Stereotactic radiosurgery is another form of radiation that's used to treat functional abnormalities and small tumors of the brain. So again, it's really focused on delivering precisely targeted radiation and in fewer treatments. So they really use high dose radiation, but in much fewer treatments for the patient. And proton beam therapy, which is able to deliver higher therapeutic doses of radiation to tumors. Again, it has that more accurate targeting of the tumors, and this gives lower radiation exposure to the normal tissue. Next slide. So in parallel, these two often get confused with one another. So while radiation therapy you can think of as a machine delivering these guided beams of radiation directly to a specific spot, radiotherapy is something very different. So radiotherapy is therapy that uses radioactive isotopes to actually kill the cancer cells. And it's a targeted therapy. So again, when we think back to those different types of therapy, this is a therapy that is usually delivered systemically, so it's usually injected into the bloodstream, but it has a very specific targeting vector, which is usually an antibody, but it can also be a small molecule or a small chemical substance that targets something very specific that's overexpressed in the tumor. So prostate is a prime example of this. Most people will have heard of the prostate-specific membrane antigen, so PSMA. This is a widely known marker for prostate cancer. So if you think of having an antibody that will bind to PSMA, PSMA is found in huge levels in prostate cancers. So we can inject that antibody, which is radio-labeled with this radioactive particle. So it means that radioactive particle is basically stuck to the antibody. We can inject it into the patient. It travels through the bloodstream until it finds PSMA. When it finds PSMA at the tumor, it binds to it. And now we have that radioactive particle that is essentially bound to the tumor. And you can think of that radioactive particle as being like a little radiation machine. It's emitting radioactivity directly from itself. It does not travel very far in the body. It depends on the isotope used, but most of them can only penetrate about 2 to 4 millimeters in tissue. So it's really focused, specific therapy. We have seen that there are people who actually inject radiotherapies directly into the tumor lesion themselves. So sometimes if you don't have that overexpression of a protein on that tumor, you might need to actually just inject it directly into the tumor, which things like melanoma and other types of cancer are pretty easily done for. But again, we really focus usually on an antibody that's radio-labeled with this isotope. Next slide. So I just want to touch for a second on something that we actually have in Toronto. So this is the Sunnybrook Cancer Ablation Therapy Program. So talking about those radiation therapies and radiotherapies that we just touched on, this is a clinic where they actually have 3 of these really highly specialized therapy machines all in one center. So they have the MR-Linac, which basically allows them to take an MRI and watch the beam of radiation move through the patient's body in real time. So they can really be very precise about where that beam is going because they can actually watch it in real time. Then they have the Gamma Knife Icon, excuse me, which allows them to, again, deliver this precision radiation to a broader group of patients. So they can give it to as many people as 30 with, to people with as many as 30 brain tumors. So it allows them to do multi-targeted radiation therapy. And then they have an MRI brachytherapy suite. So this is, brachytherapy is just what we talked about on the last slide, where we have that radiotherapy that's very specific and using radioactive isotopes. And they now can actually guide that radiotherapy as well and watch it in real time with MRI, which is amazing. Next slide. So chemotherapy, of course, everybody already knows what chemotherapy is. It's probably one of the most widely known and widely feared therapies for cancer. It's really a pretty broad term, actually. We think of it very specifically, but chemotherapy can really be any type of chemical drug that is used to kill cancer tissue. And again, like radiation, chemo is most effective on rapidly dividing cells, but it also is not specific. And so, of course, it does target normal, healthy cells as well. So chemotherapy resistance is common and is one of the most difficult challenges to using chemotherapy for patients. And for this reason, this is why people are usually given a cocktail of different types of chemo drugs to overcome the natural resistance that a patient might have to any one of those types. So I've listed a few of the different types of chemos at the bottom here. So you have agents that are DNA damaging, anti-mitotics, so they disturb the mitotic cell division cycle, anti-tumor antibiotics. We have lots of different types. These are basically just chemicals that have different types of chemical structures. So the different chemos fall into different groups based on their chemical structure and exactly how they kill the cells in the body. Next slide. So nanoparticles are what I would consider a flashy division of science, and they have previously been used for just some fancy science, but they really have started to emerge as actual very useful particles for delivering cancer therapeutics, and in particularly chemotherapy, I would say, has really benefited the most from nanoparticle advancements. So nanoparticles exist on a nanometer scale, as their name implies. They're very small, and basically they can be used for drug delivery. So we can either encapsulate a drug inside of the particle as it's being made, or we can even functionalize the cell of the particle so we can attach the drug directly to the surface. And by doing this, we can take drugs that are either hard to get into solution. So if you put sugar into your hot water, it dissolves very easily. But if you put, let's say, flour in a large quantity into your water, it's not going to dissolve. So something like the flour, we can attach it to a nanoparticle to make it dissolve better into the liquid so that we can deliver it to a patient easier. And another thing that this is used for is also there are certain chemos that are very toxic to normal tissue, and we really don't want patients to get high doses of them to their regular tissue. So if we bind them to a nanoparticle, we can actually help to get that chemo delivered directly to the tumor without letting it deposit in other areas of the body, damaging other tissues as well. Next slide. So we touched on this a little bit at the beginning. So hormonal drug therapy, this has come about for various types of cancer. Of course, breast is the most well-known. So some cancers, such as breast, are fueled by the overexpression of different hormonal biomarkers, like the estrogen receptor that we were talking about earlier. So what this means is that the more that the estrogen receptor is upregulated, the more the cancer grows. It's like food for the cancer. And when we use hormonal therapies and block these receptors, it's like starving the cancer cells. So basically they need that overexpression, and that upregulation of the estrogen receptor allows them to keep proliferating and keep growing. And you can think of it as Pac-Man, and he's coming along and eating all of those estrogen receptors. And as he eats them, they just keep filling in again behind him. So he can just keep going and eating them. But when we bring in an antibody, he eats them, but it doesn't allow them to regenerate behind him, and eventually he runs out of things to eat and it starves the tumor. Next slide. So high intensity focused ultrasound is very similar to just regular ultrasound. So I'm sure most of you have had an ultrasound in your life. So it's a really non-invasive procedure to go through. The only difference for this form of ultrasound is that they use much higher frequency sound waves. And so at this high frequency, the waves actually begin to heat up, and they kill the tumor tissue basically through direct heat as well. And this really, though, can only be used on a primary tumor. It does not induce an immune response, so it has virtually no effect on distant metastases. It's commonly used for prostate because most of the time if prostate can be caught in the early stages, we really just have this one primary focal lesion, so it works really well for prostate patients. Next slide. So microbubbles, you can think of similarly to nanoparticles. They are small vessels. So instead of just being kind of like this open particle, they actually have gas inside of them. And they're commonly used as contrast agents for imaging. So we can, again, functionalize the outside of them. We can put things inside of them. And so we can use them to kind of see ultrasound. And now, more recently, they've also started to be used as carriers for targets. So you can actually, again, use them like the nanoparticles for drug delivery by either putting the drug inside the bubble itself or attaching it to the surface. And if you can play the little video, Kim.
Oops.
Oh.
Okay.
Or maybe it's working.
I don't think it's gonna work. Nope, it's not working.
That's okay. You guys can all take a look at it.
Oops, sorry.
One that was sent out, but it's just a little video showing you the microbubbles actually working. So you can take a look at that and just email me if you have any questions. Next slide. So stem cell transplants, these of course are typically used for patients with blood cancers. So we take the patient, we want to, excuse me, we want to give them high dose chemo or different types of treatments to suppress their immune cell population. Then we harvest their blood and isolate stem cells. Then we give the chemo to remove basically the bad immune system in their body and depress that bone marrow before we now give them the transplant with the fresh immune cells. And this helps them to have a better response to drugs, actually, because now they have viable immune cells in their body that can actually fight these cancer cells. And really, stem cell transplants, like I said, have primarily been used for liquid cancers. Interestingly, they also were used for quite some time for breast patients and for some other types of cancer as well, where they thought that by doing these transplants, they could actually cure patients. But of course, we all know that's not used today, and they really only have seen efficacy in liquid tumors. And of course, one of the major issues that comes with stem cell transplants that really is something that is still heavily researched to this day, of course, is graft versus host disease. So making sure that the new immune system that's put into your body does not actually attack your body. And, you know, there's this constant battle of trying to discern whether or not this can be integrated into your body as your own immune system and not recognized as a foreign entity. So that's, of course, always one of the problems that comes up when we talk about transplants. Next slide. So genomic profiling is a more recent approach to cancer, and it's something that falls really into the category of personalized medicine as well. So I think that I'm sure any of you that have had cancer or had a loved one who had cancer know that it's pretty common nowadays to get genetic testing done and to see a genetic counselor. And there is a really wide array of mutations nowadays that we are pretty certain about and know a lot about. But genomic profiling goes even deeper than that. So really, we're coming into the era of personalized medicine where I think in the future and maybe in the next 10 years, we'll see genomic profiling of a patient's entire genome becoming much more prevalent. For now, it's a pretty lengthy and slow process, and it's also expensive. And so unfortunately, it's not put into regular rotation right now, but it is something that allows us to really look at a patient's individual genomic landscape and see all of the mutations that they have and all of the changes they have and what things are upregulated in their tumor and what things are downregulated. And this really helps us to pick exactly what treatments are going to help that patient. And so I encourage you all to push for genetic testing. It's something that is still a little bit controversial. I think they'll give you a standard gene set of tests usually to start, but you can always ask them for more. And it's really no harm to you. And I think it really gives a lot of information to the scientific community. And it also gives a lot of information to you, because as you start to see more research coming out in the next decade, I think it will really help you to understand your own cancer and your own treatments, knowing your genomic status and what mutations you have and what the landscape of your tumor was. And this is really going to be a big area moving forward in cancer research. Next slide. So Watson for Oncology is thought of as being an AI system for cancer treatments. It's not actually AI. It's actually it's a machine learning system. So it's a little bit different than AI because it's trained by data. So it's not given rules where the neural network kind of builds its own ideas. It's just a machine learning system. So it really takes all of these data sets that have been put into it and basically runs all of these statistics so that when a clinician comes and has a patient that they present with, it can help them to just devise an individual cancer treatment for their patient. So it's a really unique and cool program because it kind of allows an oncologist to be able to have all of the data sets at their fingertips to help them make an informed decision for their patient, whereas the oncologist otherwise would usually be too busy and probably too out of touch a lot of the times with all of the literature to be able to have that knowledge otherwise. Next slide. So this is also a video. I don't think it's going to work then.
No, I think it's because we're on Skype, so
Okay.
apologies for that.
No worries. So you guys can take a look at this video on your own. It's a really nice video, just kind of showing you what immunotherapy is. So immunotherapy is really, really big in the field right now. In 2018, we had 2 scientists win the Nobel Prize for discovering 2 different pathways for checkpoint blockade antibodies, which we'll talk about in a minute. And that's a type of immunotherapy. So immunotherapy and this whole concept of the immune system being able to fight the cancer within you was originally proposed back at the beginning of the 1900s, like we said, but it really is now, the era of immunotherapy. And really we're talking about a bunch of different types of therapy, and they all just have this common principle of enabling these immune cells to get into the tumor and to fight the cancer tissue. Next slide.
Yep.
So I'm going to go through some different types of immunotherapies because there are many different types. So we're going to start with oncolytic viruses. So oncolytic viruses, interestingly enough, actually came from clinical observations. So basically doctors saw that natural human virus infections led to the regression of tumors. So they just had a patient who had a tumor, they got a natural viral infection, and that viral infection actually cured their tumor. And this really opened the door for researchers to the idea that we could now harness these viruses and actually use them as viable therapeutic options for cancer patients, which was a really mind-blowing idea at the time. So oncolytic viruses preferentially infect and kill cancer cells, so there's usually very little toxicity or side effects to the patient. And over time, we've also learned that we can actually further manipulate the genetic code of viruses. So we can actually change and clone things into the genetic code of viruses to make them better at killing cancer and to decrease any toxicities that they might have to healthy tissue in the patient. And this is a really hot topic right now, and there's a lot of people working on a lot of different oncolytic viruses. There's only one currently approved by the FDA. I actually don't know if it's used in Canada, to be honest, at the current time. It's called TVEC. It's a herpes simplex virus, and it's approved for actually first-line treatment now for melanoma patients, and it works very well for them. We've also seen it used, I believe, for non-small cell lung cancer, and it's actively being looked at for various types of cancer. So oncolytic viruses don't really have a preference, I would say, in terms of their research, where they're being used right now. I would just say they're more being used for solid tumor types. Next slide. So checkpoint blocking antibodies. So this is what I was talking about for the Nobel Prize. So basically, we have these immune cells in our body called T cells. So there are different types of T cells, but all we need to think about is that T cells are the cells in your body that can kill damaged or malignant cells. So they can recognize these cells as abnormal, and they can start the process to signal for apoptosis to have these cells destroyed and removed from the body. So T cells themselves, if we look at the picture here, this is a T cell on the left. T cells have tons of different pathways with all of these receptors attached, sticking out of the cell. So they have regulatory pathways, and they have these inhibitory pathways. And the inhibitory pathways can basically stop the T cell from working. So it can turn off its killing properties. And what these two researchers discovered separately, actually, is that there are two pathways, CTLA4 and PD1, that I have the boxes around there. And these pathways are actually upregulated in a lot of different cancers. And if we use an antibody to come and bind to those receptors, we can actually shut down those pathways. And a lot of the times when we shut down those pathways, the T cells are now able to kill the tumor when they previously were not able to. So this is this was an astounding discovery. This has been something that has been really the focus of literature for the last few years. There are so many clinical trials going on right now with various checkpoint antibodies. Most of them are focused on these two pathways, CTLA4 or PD1, and basically it's just an antibody delivery for the patient. But again, this really depends on the amount of upregulation. So when I talk about that genomic profiling and that personalized medicine, checkpoint works best in a patient that already has high levels of these things in their tumor. So if you have high levels of PDL1 and PDL2 in your tumor, that means that you are likely going to respond very well to anti-PDL1 checkpoint blockade therapy. However, if you don't, you may not respond at all. So this is part of the problem, and this is where a lot of the research is right now, is really focusing on, okay, we have patients who naturally have the right tumor landscape to respond to this therapy, but for the patients who don't, how can we change the landscape of their tumor so that they also can benefit from this therapy? And that is a massive area of research right now, and there's a lot of funding in that right now. Next slide. So CAR T-cell therapy is another type of immunotherapy. This is very similar to what the next slide is as well, which is adoptive T-cell therapy. So I'm going to kind of lump them a little bit together. But basically what we're talking about when we talk about these T-cell therapies is we're talking about this process where we take a patient's blood, we isolate the T cells from the blood, then we modify those cells. So we can change them to help them to better recognize and target the cancer. So we can literally do this. If you remember from the Cancer Biology 101 webinar where I showed you, excuse me, how we actually look at cancer, and I showed you those flasks and dishes where we actually grow the cancer inside of that media, that's literally what we do with these patients' T cells. So we literally grow them inside of a growth media that has all the nutrients they need to survive and keep growing. And then we expand them. So after we've engineered them and made them better at killing the cancer, we grow them up and make as many as we need to then infuse them back into the patient and have them now fight the cancer themselves. And this therapy works very well, but unfortunately there have been some toxicities associated with it. And that's really the biggest driver in the research for T-cell therapy right now, is limiting the toxicities and how can we make it even better. So in Canada, actually, CAR T-cell trials are not in existence. You actually, I don't believe, can even get CAR T-cell therapy here right now. But in the States, the amount of trials are endless right now for CAR T-cell therapy. So it's definitely going to start emerging in Canada soon in the near future. There is a professor at McMaster who's actively working on this and moving into clinical trial phase. So this is something that we're going to see a lot more of in the next few years. Next slide. So adoptive T-cell therapy is the exact same idea. It's just the natural T cells itself, whereas CAR T therapy actually attaches itself to a receptor on the T cells. So we're really talking about two very similar things. Again, really just looking at T cell therapies where we're engineering the T cells to better recognize the cancer, growing them up into a large population, and then introducing them back into the patient to kill the cancer cells. Next slide. Oh, so this video is not going to work. But so CRISPR is also a very big area of medicine right now. So it is something that you'll probably hear a lot of at the conference. So basically, CRISPR is something that we use to edit genomes. So it allows us to actually alter DNA sequences and modify gene functions. And you've mostly heard about it in the news because it comes with, unfortunately, some moral issues. And, you know, it can be used for various types of cloning, and there are moral and ethical dilemmas and questions that arise when we talk about that. But for cancer, CRISPR is actually a very useful tool that has allowed us to edit genes within organisms. It really has hugely widespread application. Next slide. So how is it used for cancer therapy? So CRISPR can be used as a screen. So we can actually do these things called CRISPR screens where we look at huge numbers of gene sets, and we basically use it to identify new therapeutic targets for cancer. It can also be used to create genetically modified mice. So we can actually change the genome of mice. We talk about it as, you know, either knocking in or knocking out certain genes. So we can either insert genes or remove genes. And basically, this is just a tool that allows us to do this very easily so that we can now study if a patient is mutant in this gene or is lacking in this gene, what happens? If we impair the function of this gene, what happens? You know, is this gene necessary for the apoptotic pathway for cell death to occur? So it really allows us to get into the nitty-gritty of the mechanistic work behind cancer and really start to identify, like I said, new targets, but also how different genes come into play. We also have been able to use it now to manipulate those T cells for T cell therapy. So we can basically use CRISPR. You can think of CRISPR as like a little delivery tool. It basically delivers these little sets of DNA, excuse me, into a genome. So we can actually really easily use this now to manipulate your T cells when we take them from a patient to put in exactly what we want so that they can better recognize and kill those cancer cells. And just the screenshot on the left there is a very small portion of a lot of clinical trials that are currently running that use CRISPR. So it is actively being used in the clinic, and it's something that I think we're going to see more of. And of course, like I said, there is that moral and ethical concern, but I think that is really when we talk outside of the realm of cancer. I think, you know, using CRISPR for other purposes has much more moral complications, but for cancer, it's actually a very useful tool that's being actively used in research laboratories and in clinical practice. Next slide. So vaccine therapy. So if you pay attention to the news, you will regularly see people making pretty crazy claims about cancer vaccines and how we're going to have one vaccine for all of cancer. And of course, that is really not realistic and is not actually going to happen. But vaccine therapy can be used. So we have two different types. We either have prevention vaccines, which would be like the HPV vaccine that we talked about earlier, or we can have cancer treatment vaccines. These are much more difficult to identify and make, but they are possible. So we can basically give them to prevent the cancer from coming back. or to destroy any cancer cells that are still in the body after your other treatments have ended. So we have these things in our body called antigens. These are basically foreign substances that come into the body as a response to the cancer. And these antigens are usually recognized by the immune system, attacked, and cleared. So they're recognized as being something foreign. The immune system attacks them and clears them from the body. And this leaves the immune system with a memory. So it remembers that it recognized those specific antigens. And when they come back in the body in the future, it recognizes them because you have that memory phenotype in your immune system. So cancer treatment vaccines boost the immune system's ability to recognize and destroy cancer cells that have these specific antigens on their surface. And some can be made for the individual patient and are actually produced from their tumor samples. So an example would be that men with metastatic prostate cancer actually have a cancer vaccine called Provenge. And again, we're really just talking about something that is developed to recognize the antigens within the tumor, so the foreign substances within a tumor, so that instead of just getting a debulking effect of that tumor, it doesn't matter where those cells are in the body. You could have one cell floating somewhere very distant in the body, but if it has that specific antigen on it, it will be recognized and destroyed from the body. And right now, there's actually tons of clinical trials going on for vaccine therapy development across many different types of cancer. And it is an area that is widely studied in research labs right now as well. Next slide. So complementary therapies, we should maybe title this like complementary strategies instead, but we're really talking about things such as, you know, functional foods and oral supplements, homeopathy, naturopathy, yoga, massage. So these are all things that are strategies that have been widely marketed. And, you know, you'll see an article almost daily, I'm sure, about how blueberries will cure your cancer. And that's not true, of course. And unfortunately, most of these things don't have scientific evidence to support them. So it's really important to recognize that these complementary therapies do not treat the cancer itself. So the purpose is to help improve your overall health and well-being, and they can really help patients to cope physically and emotionally with the conventional cancer therapies that they're getting. So, for example, patients who have chemo, obviously, even with the wonderful anti-nausea drugs that we have nowadays, they have a lot of nausea from the chemotherapy. And having something like acupuncture can really help control that nausea and help them to feel a lot better and improve their quality of life. So that's an instance where this is a complementary strategy that really helps a patient who's going through chemo, but it's not in itself treating the cancer. Excuse me. On that note, there is a lot of research that does suggest that certain complementary therapies can help because they can play a role in things like influencing your gut microbiota. And this is a new role, a new area, I would say, in cancer research, where there is actively a lot of people looking into your gut microbiota and how that plays a role and how you respond to conventional therapies. So again, it's not looking at how these things directly kill cancer, but how they can play a role in how you respond to the other cancer therapies you may be receiving. Next slide. So I think this is the last slide. Just to finish, I just wanted to really touch on combination therapy. So while I've shown you all of these different types of therapies, clinicians ultimately want to treat their patients with the fewest treatments that will have the most impactful outcome. So ideally, a clinician is wanting to give their patient one drug that's going to cure their disease. But that's not realistic, unfortunately, for cancer. And monotherapies often do not work and have very little efficacy for patients. And that's why we really have to focus on combination therapies to target the heterogeneity of tumors and overcome that acquired drug resistance, unfortunately, that patients naturally have to things like chemotherapy. So we really have to take all of these different therapies together and we come into some unique combinatorial regimen for that specific patient that could help them overcome their disease. even when we talk about new emerging immunotherapies that are coming into the clinic and coming down the pipeline, most of these are given in combination with either another type of immunotherapy or with chemotherapy or with radiation or with surgery. So it's really finding this unique combination that's going to work for a patient, that's going to change the tumor microenvironment just enough to allow them to really respond to those efficacious therapies and have the best overall survival and best prognostic outcome that they can. Next slide. So with that, I'll thank you guys for listening. That was a lot of information, I know, and didn't go into huge detail in each of them, but you can always email me if you have any questions at all, or if you want to just chat about some of the stuff we talked about, or if you have questions. If it's something that I am not an expert on, I can always direct you to someone who is more of an expert on that, and they can help answer your questions as well. And I think we do have about 10 minutes still if you guys do want to ask any questions now.